Hi, welcome to a physionic detailed study analysis. Today we're going to be going over 5 different studies, one of which being a meta-analysis, including more studies than the 5 that we'll be going over. And we're going to try to determine the effect that creatine supplementation has on our memory. But beyond that, we're also going to be going over some of the other neurophysiological effects of creatine. Now, this is a lengthy, detailed study analysis, so if you'd like to skip to the conclusions, please feel free. However, you will be missing out on considerable context. However, I've grouped all the highlights into 1 or 2 sentences at the end of the analysis for your convenience. For those nerds that would like to continue onwards, well, if you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, hold my master's in exercise physiology. I've actually written a book on creatine, and this is what I do. I take studies or groupings of studies and try to come to some form of consensus, some form of understanding. That doesn't mean that you have to agree with my conclusion, but I'm going to present the data and then you can make your own mind up. Okay, with that out of the way, with that introduction out of the way, let's go ahead and jump into this. So the topics that I'm going to be covering for you are the mechanisms of action. So what are the proposed mechanisms that creatine has on our brain? Then we're actually going to look at the real world effects of creatine on our memory. Then I'm going to really intriguing difference, look at potential differences between men and women. And then we're going to look at an age effect, if being older or being younger, if there's some sort of an effect there. And then if you're part of my insider program, you'll also get access to the dose type, so the way of consumption, and some other really intriguing nuances when it comes to creatine supplementation. Then creatine on sleep deprivation and stressful situations, and some of the other neurological effects that could occur from creatine supplementation. If you're interested in having access to the full version, as well as a whole host of other benefits that are included in the Insiders program, along with an Insiders podcast, including other study breakdowns, research reviews, and a whole host of different things, live sessions with me, then certainly you're welcome to join the Physionic Insiders program. But if you're interested in just the main outcomes here, so looking at creatine's memory effect, then let's jump into this. So the mechanism of action. So let's get into the cells. Let's, as we do in Physionic, let's dive into the intricacies of the cell. So we've got our brain here, and let's say we were to look at a particular neuron, so just one brain cell, and then we were to zoom into that brain cell and we were to look at just the cell membrane. Here we've got our neural cell membrane, got the outside here, and we've got the inside here. So this is the cytosol, and this would be the extracellular fluid. Now, inside your cells, you have ATP. You have these molecules of adenosine triphosphate, and they are known as the currency of the cell. They are the energy currency of the cell. Now, there are other molecules as well, for example, like GTP, which are also energy currencies of the cell, but the main focus for a lot of biology is focused on ATP specifically because there's so many reactions, so many more reactions that occur with ATP. So, how do we generate ATP? Well, we can go through a normal process known as glycolysis, which is taking glucose molecules from outside the cell. They enter inside the cell, and then they go through the glycolysis pathway. So they get this glucose molecule gets acted on by a series of different molecules, enzymes, and those change the structure of glucose. But as they do that, they start to spit off and start producing ATP. Now, additionally, glycolysis can go into the mitochondrion through a process called aerobic glycolysis, which can ultimately lead to more ATP than just anaerobic glycolysis. So, but the point is that it can go through this metabolic pathway called glycolysis and can ultimately add to the pool of ATP that's available within the cell. Now, beyond that, however, there are other molecules that can enter the mitochondrion. So like astrocyte metabolites. Now, the reason why I'm pointing this out is there's a very special distinction between neurons and some of your other cells, the majority of your other cells. If you were to take a muscle cell, if you were to take a liver cell, for example, it would be able to take up fat molecules and then be able to put those in the mitochondria and generate ATP. However, your neurons have a very limited to no capacity to do that. So what they do is they rely on other cells called astrocytes to take up those fats, to generate those metabolites, which would generally be made by the cell itself inside the mitochondrion or just outside the mitochondrion. And then because it's relying on these metabolites, these astrocytes have to kick those metabolites out. It's almost like you can think of it like a bird pre-chewing the food for its babies. The neuron is the baby, and the adult bird is the astrocyte. So the astrocyte is supplying the chewed up food. So the chewed up fatty acids in this situation, the chewed up fat molecules in this situation, and turning them into NADH or other energy producing metabolites, which ultimately get exported out of the astrocyte and then enter inside the neuron itself. And then those can then go into the mitochondria and generate ATP. So it's a very symbiotic relationship between the two, although the neuron's a little bit more selfish than the astrocytes. The astrocytes are very giving. Okay, so what do we use ATP for inside our brain? Well, there are a whole host of different functions for our brain. However, one of them is in the exchange of ions across the cell membrane. So again, this is the outside of the cell, this is the inside of the cell. So ATP is involved in the sodium-potassium pump. So pumping sodium back out of the cell from the inside to the outside of the cell, and allowing potassium back into the cell. And we have different leak channels as well, but for the most part, when we're talking about ATP intensive processes that happen within our cell, this is one of the major ones. And this is the one that I'm going to highlight specifically for this conversation when we're talking about creatine. So the point is that you usually have a high gradient of sodium outside of the cell and a relatively larger gradient of potassium inside the cell. When you look at the absolute values, it's a different story, but as it stands, without getting too confusing, just know that there's more sodium outside the cell. There's more sodium outside the cell and more potassium inside the cell. So the cell has to continuously make sure that there's the right amounts of both on either side of this cell membrane. And it does that through this potassium pump. Now, or sodium potassium pump. And as such, it uses large quantities of ATP to facilitate that function. So to give you an example here, so we've got a neuron here and we've got a neuron here. And when they're in their quiet state, so they're not activated, so they are in their quiescent state, then you have the sodium that's outside. You've got a sodium channel here, and you've got a potassium channel here, and you've got potassium that's found inside of the cells. We've got our cell membrane, outside the cell, inside the cell. Now, for a signal to propagate and to move all the way down this first neuron and then to communicate that signal across neurons from one cell to the other cell, you have to have this mass exchange inside this neuron of these electrolytes of sodium and potassium. So suddenly sodium will rush into the inside of the cell. This changes the chemistry of the cell itself to the point where you have this reaction. And this is, I'm being very general here because that's not the point of this conversation. The point is that just all you need to know is that there's a reaction, an activation of the cell when sodium enters the cell. Okay, that's all you need to know. And what also tends to happen is the potassium is then effluxed out of the cell. So this whole process is called depolarization, repolarization, and hyperpolarization. Those words themselves, if you're in biology, you will recognize them. If you're not in biology, you may not recognize them. It doesn't matter. The point is this whole process, this exchange, this reversal of from the quiescent state, remember the quiescent state was with the sodium outside, the potassium inside, this reversal where sodium is suddenly inside the cell and potassium is now outside of the cell in very relative basic terms. So there's a lot more specificity here. I just have to be clear. Then that leads from a quiescent state to an activated state. That activated state means that there's a signal that gets propagated all the way down the cell and then eventually gets carried over to this next cell and then activates that cell. So how does the cell then return back to its quiescent state? The way that it does that is by these sodium-potassium pumps and through these leak channels. So the sodium will then be kicked back out of the cell and the potassium will then be allowed back into the cell. And this is an energy-intensive process. And if this has to happen millions and millions and billions of times as each neuron is communicating with another neuron to essentially form memories and to form different neurophysiological functions for the brain, then this is an intensive area of energy use. Okay. So let's return back to this. Nothing has changed here. We've got ATP that's very involved in the sodium potassium pump. How does this relate to creatine? Well, it relates to creatine because the creatine molecule can actually take up what's known as a phosphate. Remember, I called this the adenosine triphosphate, meaning that it is a molecule that is an adenosine molecule plus the addition of 3 phosphate molecules tagged onto it. Now, that is a fully charged ATP molecule, and that can then be used for cellular energy. So what happens when you use the ATP? Well, the ATP gets degraded or gets cleaved, gets changed from an ATP to an ADP. So adenosine triphosphate, 3 phosphates attached to an adenosine molecule, to adenosine diphosphate, so 2 phosphates. So you lose a phosphate. Guess what creatine does? Creatine can be phosphorylated by an enzyme called creatine kinase, which ultimately leads creatine molecules to now be turned to phosphocreatine or creatine phosphate, but usually people say phosphocreatine. So this phosphocreatine, guess what that does? Because it has a single phosphate on it, what do you think it can do to ADP molecules? It can re-energize them. You can offer, the creatine molecule can essentially offer its phosphate to the ATP and re-energize it back to an ATP. So therefore you get the recycling of energy. And that's the beauty of creatine, that you don't have to go through glycolysis to generate new ATP. You don't have to go through the mitochondria to generate more ATP, which would require more substrate. You can just use creatine as a tertiary system to generate more energy. Now, if you have more energy, what do you think happens here? That means then you have more energy available to have this quick communication between these cells, between these neurons. And that ultimately leads to better overall neurophysiology function. Now, that is the canonical system, and that is only a simplistic, that's the major system, but there are other mechanisms by which creatine can also have these effects. I'm not going to be going into them here, but just as a few things, creatine can change gene expression within the cell, can lead to greater expression of particular proteins that would be pro-memory. It can also act as an antioxidant. It can also protect mitochondria by globbing onto mitochondria and making sure that they don't end up bursting. There's a number of different kind of less known mechanisms that creatine can have an effect on the brain. But as it stands, this is the primary one. So it's an energy-driven effect. Now, let's talk about creatine on memory. What are the actual real world effects? It's interesting to go into mechanisms. I always love going into mechanisms. I always make sure to have that for these study analyses so that you can understand why it's actually having an effect, if there's an effect. But it's also important to look at the data itself. So we're going to be relying a lot on this meta-analysis. So this meta-analysis, which I've labeled Study 166, did not have any funding. So there's no external funding of any sort. I don't even think there's any internal funding. It's just a group of researchers that are aggregating the data across a number of studies. In this situation, we're talking about 8 studies that were included in the meta-analysis. You can see their rubric here for how they ended up accepting certain studies. So they had exclusion and inclusion criteria, which are right here. So the inclusion criteria had to be in healthy adults, had to be creatine monohydrate supplementation. So we're not talking about creatine, some other forms of creatine. like creatine HCl as one example. The comparator had to be a placebo, so it couldn't be like omega-3s or something like that. It had to be a placebo. And they were looking for memory performance outcomes. And this was restricted to randomized controlled trials. So we're talking about actual cause and effect kind of studies. Now, the exclusion criteria would be obviously in many instances the opposite. So non-randomized controlled trials, cohort studies, et cetera, in vitro studies, so talking about cell studies, looking at other outcomes related to cognitive function. So maybe reaction time is another example, which we'll be getting into if you're part of the Insiders program. I'll be going into a little bit on reaction time and executive function and a number of other brain indices. But they end up excluding those in this analysis. Non-placebo control. So if it doesn't have a placebo in the study, then they exclude that. So if I can go back here real quick. So they're excluding certain studies because of that, ineligible outcomes, incompatible populations, maybe populations with comorbidities or dietary restrictions. Maybe it was just in vegan individuals. So that was part of their exclusion criteria, so they removed that. Okay, so what did they find? I should note here that these researchers had to correct their analysis. Initially, they had a particular analysis that included many, many more of different outcomes. However, one of the drawbacks of that was that it is statistically improper to have multiple measures of memory coming from the same sample. What do I mean by that? I mean that if you are going to do an analysis, a meta-analysis of this sort, you have to make sure that each one of these data points, each one of these studies has a unique sample, so a unique group of individuals that were studied. You can't report multiple measures of, let's say, memory. So let's say they do test A, test B, test C on one sample, one group of individuals, because that's bolstering the data without actually having unique individuals involved in the data collection. Hopefully that makes some sense. It's a little difficult to explain, but the way it's supposed to work is that you have one sample, you're allowed to take one measurement and use that one measurement in the meta-analysis. If you have one sample and you take multiple measurements, then you are artificially kind of inflating the results of the analysis. That's the best I can do on the fly for an explanation, so excuse me. So they ultimately at first indicated that there was a benefit of creatine supplementation on memory, just general average memory. However, when they got called out by other researchers for this significant statistical error, they ultimately redid the analysis, and that's what I'm showing you. So I'm ignoring the data where they did a lot of these repeat measures across multiple test criteria, or these multiple tests in general. So this is all the studies, and they took one test from each sample, and ultimately what did they find? Well, to read this, I'm not going to go into all the details of how to read this and whatnot, but these are all the studies listed out. Here's the mean under the creatine group and the mean under the placebo group. They take the difference between that, so standard mean difference, so SMD, and then they end up reporting it. The actual visual of all these numbers here is listed right here. So the green is the indication of the mean, so the actual average result that they ended up finding, and the lines on either end are the confidence intervals, so how certain are the results around this mean. And ultimately, this line down the middle is 0, indicating that there's no effect. If all the studies move more towards the left, then that means that there is a harmful effect of creatine on memory. And if the results move more towards the right, then that means that there's a benefit of creatine on memory. So that's because the placebo, in favor of the placebo, would move to the left, which would then mean a detriment to creatine. And if it moves to the right, then that's in favor of creatine, as we see here. So it seems like if you were to look at this, if you were to aggregate all this data together, and then they do that by putting that into this black diamond. This black diamond is taking all of the results of all these studies, all these numbers, and just aggregating it into one really easy way to just look at the average result across all the studies. And what you would think is that even though you have a few studies that maybe lean a little bit more to the left, you have one study here, this Rawson 2008 study, that indicates there's essentially no effect. Maybe the same could also be said of this study. The Merej-Filho study would also say that there's maybe no effect, maybe a slight detriment to creatine. But you also have a few other studies that indicate that are strongly in favor of creatine. So in aggregate, what does this tell us? Well, if we look at the diamond, it seems to move a little bit to the right in favor of creatine. However, if we actually don't trust our eyes and we use the statistical measures here, we indicate that actually there is no statistically significant effect. So they went from a position of, yes, there is a benefit of creatine, to technically now there's no benefit of creatine. And that's after they've removed all of those confounding measures that they did in their initial analysis. So with a p-value of 0.15, it's starting to maybe trend towards that direction. And maybe if they had more data, they might be able to get, which clearly they did, but obviously they had to remove it. So more unique data, I should say, then maybe they might see an effect. But as it stands, there does not seem to be an effect of creatine supplementation on the brain. So the next question we have to ask ourselves is there an age effect? So it's one thing to look at the overall effects and then say, okay, well, then I guess we can ho-hum, we can just go home. But in reality, analyses in science is a lot more sophisticated than that. And we can actually do what are known as subgroup analyses. So not only look at the overall effect, but start to break out studies and try to separate out studies based off of particular characteristics. In this situation, we're talking about age. So it's the exact same read. We're doing the exact same thing, but we've broken it up into two different groups. One group is younger adults and children, which they've listed all the studies there. And then they've got older individuals. They've got two studies here. So now we're breaking it up by age, and the age cutoff point is 65, if I remember correctly. So if we look at the younger adults and children, we see that all these studies are relatively in line with one another. And we can see that the diamond is smack dab in the middle, indicating that there's no benefit or harm of creatine supplementation. Now, if we look at the p-value, there's a strong indication that that is correct. So the interpretation, what we would see here, what I would see at least, I'm only speaking for myself, that would indicate that there's no effect of creatine supplementation in younger individuals. Now, on the other hand, for older individuals, what we see is that a strong shift towards the right. And remember, what does that mean? Well, that's in favor of creatine. So it seems like the overall effects diamond is moving towards the right. Now, if we check the statistics, what does it show? It shows a strong p-value of 0.004, indicating that's likely a real effect. So it would be nice if they had a few more studies in here, but as it stands, these two studies strongly say that for older individuals, those over the age of 65, that creatine supplementation is a benefit to memory. So think about that. What a remarkable discovery to go from, creatine has no effect, to now breaking it up by age, and now you're actually seeing an effect. And that's where those nuances are incredibly important. So for older individuals, there is an effect. Now, what about differences between men and women? So here what they did is we've got males and females together. Okay, then we've got the separation of just females, and then we have the separation of just males. Now, for males, we only have one study that's specifically only males, as opposed to samples that include maybe 15 men and 10 women, or 20 women and 11 men. So those would be the females and males. And females studies is just women. So, maybe 10 women and no men, obviously, or 20 women and no men. So because of that granularity, now we can actually try to aggregate the data and try to figure out, okay, well, if we separate the data based off of those groupings, do we see an effect just like we did with age? So in the combination of men and women, we see that there's no effect. So it slightly leans towards the right, but if we check the statistics, it's a p-value of 0.36. So there's no effect. There's a little bit of heterogeneity for anybody that's interested in statistics. And that's probably just driven by this McMorris 2007B test, or study, I should say. And it's still not very high, though. Now, for women, again, there's only 2 studies in this situation, but it seems like with these 2 studies, it's starting to trend towards maybe there is an effect. But unfortunately, the p-value indicates that there's no effect. So the proper conclusion, also with no heterogeneity, the proper conclusion is that there's no effect in women specifically. And then when we look at men only, of course, there's going to be 0 heterogeneity because it's not applicable. There's only 1 study, so we can't compare between studies because there's only 1 study. You can't compare against itself. And for this, we see that the effect is there's no effect. Now, one question that might be crossing your mind is, well, okay, we're looking at men and women together, we're looking at women separately, and we're looking at men separately. Why do we suddenly see no effect when earlier we did see an effect with the age? Because obviously these have to be in men and women, right? Well, the reason for that is because here they're separating out based off of age, and here they're not doing that separation. So they're mixing together all the young individuals with the older individuals all grouped together. So that ultimately dilutes the effect that we saw up here with the older individuals. That's the reason why. Okay, so the midpoint conclusion at this point is that creatine is a benefit to memory in older individuals, those who are 65 or older, but does not seem to be a benefit for younger individuals. There's likely a need for more studies, however, and probably no real difference between men and women, but we definitely need, we absolutely need more studies in that regard. Okay, and for the insiders, let's go into dosing creatine appropriately, additional considerations, and some really, really intriguing studies on anti-stress effects and in sleep deprivation studies. So let's start with the sleep deprivation studies, actually. So we're going to be looking at this study, which is study 167. I'm going to give you a bit of breakdown on how they went about this. They had 20 men and women, which was mostly men. It was like 75% men or 80% men, something like that. They were 21 years old, so we are not talking about an age population. Please keep that in mind because that's going to be important. They were normal weight and otherwise generally healthy. They had no details on if it was randomized or not, so that's why I've got this shaded yellow because we don't know. It was placebo-controlled and it was double-blind, so mostly positive, but there may be some potential error in there. The study lasted for 7 days where the individuals were supplementing with creatine for 7 days, and then they actually underwent the experimental period. This is a parallel design, so we've got a creatine group and we've got a placebo group. Now, this is the breakdown of the overall study. So remember, they were supplementing for 7 days. Then after that, they went through the experimental period. The experimental period was a 36-hour period, if I remember correctly, where they were sleep deprived. So they went into the lab, and then the researchers made sure that they didn't fall asleep. And for that, they went through a number of light exercise, just a little bit of cardio to keep them, to keep them invigorated instead of sitting on the couch and watching Netflix where they might definitely fall asleep. Also, they didn't want to be giving them a bunch of caffeine or anything like that. So they just fed them a little bit of food and also made sure that they were active every hour. As you can see, all the way throughout this, for most of it, they just did a little bit of 15 minutes of cycling at 50% heart rate max. It's a very low heart rate max, so it's very lackadaisical. And then they do a little bit higher at hour 6, and they also take blood samples all throughout. Okay, so I found this incredibly confusing in terms of how to read this. Unfortunately, they don't actually indicate very well the statistical comparisons, which is incredibly unfortunate. So I'm going to do my best to kind of walk us through a little bit of this, but ultimately the main effect that was actually seen to be different was in adjacency. And adjacency is a measure of working memory. So the lower it is, the better one's working memory. I am not that familiar with these kinds of tests, so you'll have to excuse me. But as it stands, and this is a representation of what we end up finding up here, so adjacency here. And then we've got a bunch of other measures as well. So looking at RMG and adjacency. Adjacency is apparently also a quality control for the RMG test, and they're both working memory tests. And then CRT is reaction time, and then we've got a recall, so different types of recall. And ultimately, what they found is that, at least statistically speaking, what they actually made sure to indicate, and now that I see it, it's only 24 hours of sleep deprivation, just so we're clear, because I said 36 earlier. And what we find is that in terms of the working memory, we do see an improvement, we see a better result in the creatine group. Now, it's difficult for me to read this because, again, they just don't make things very clear. I even wrote in my notes here, this is a graphical description of Table 1. Thank goodness, because Table 1 is horrifically described. So clearly I was not pleased when I read this study, and I still remember it. We also didn't see any major changes in reaction time. Maybe at the 24-hour mark, you might argue that there's a little bit faster reaction time in the creatine group. So it's not like creatine necessarily improves the reaction time and the adjacency or the working memory effects. What it actually does is it more so protects against the degradation of working memory and reaction time and verbal recall and things of that nature. So here we're looking at reaction time, and here we've got a verbal recall. So this is a memory test, and here we've got, and this is the overall delta. So now we're looking at not just the absolute values, but we're looking at the delta values, so the overall difference from baseline versus 6 hours later, 12 hours later, 24 hours later. And here we've got reaction time. So they're two different tests. And overall, we can see that there's the recall is typically a little bit different for the creatine group compared to the placebo group. In terms, and again, was very poorly described, so it's difficult for me to really parse out. However, in terms of reaction time, as we pointed out earlier, right here with the reaction time, the CRT, we see that with the creatine and placebo groups, they're about equivalent at the 6-hour time period, which of course that's not sleep deprivation. Then at the 12-hour time period, they're technically still the same. However, you're starting to maybe start seeing the differences there. And then at the 24-hour time period, where you really are starting to get to sleep deprivation, you see that the creatine group is able to maintain or even potentially better its reaction time. Now, one of the potential confounders to compare the creatine group versus the creatine group here, which they technically didn't do, they're comparing these two, is a bias of continuously doing the same test. So the first time that you do it, you're going to be really bad at it because you don't know how to do it. And usually they'll control for that by having one practice round, and then they'll have you actually do the test. And usually they would want to separate that out by at least a day or some amount of time where the person is distracted, doing other things, as opposed to doing it immediately afterwards again. So there's a possible confounding variable in here, but I think that's probably pretty weak. And I think that probably there's still an effect of creatine supplementation. Now, again, I wish I could talk more about this verbal recall and spatial recall forward and backwards. Again, it's just like different number tests and letter tests, word tests that they're doing in the forward direction and then the recall in the reverse direction. It's just, I mean, certainly I'm not an expert in these kinds of tests, but also ultimately they're just trying to find different ways of testing a person's, you could call it quote unquote short-term memory. Now, there's some debate. I have my bachelor's degree in psychology, and I remember the researchers that I used to work with would debate about if short-term memory is actually a thing, or if it's a lot more gray, if there's a lot more crossover between the different types of short-term memory and long-term memory, if it's more of just they're all kind of lumped together. And again, they make it very difficult to actually interpret this data, unfortunately. But as it stands, the way that they were talking about it, they said that there was improvements with the creatine group. Now looking at mood, which is really intriguing. So here we've got all these different measures: fatigue, anger, tension, so like stress level, depression, and vigor. And we see lower, well, obviously for depression, tension, anger, and fatigue, you would want lower values and you want higher values for the vigor. And unfortunately, again, here, they're not actually giving us any indication of what's statistically significant between these comparisons, or what's actually being compared. If it's the 24-hour creatine group versus 0-hour creatine group, or if it's within each time period. My guess is it's between each time period. So and here we've got reaction time again, if I remember correctly. Nope, excuse me, this is fatigue. I just looked at my notes. So this is fatigue levels. So we're looking at this measure up here, but you can also look at some of these other values. So here's the only one that we actually have any indication of statistical significance. And we see that with the creatine group that the fatigue levels were lower. It did increase. It seems like it increased, although we don't actually have a comparison here. We don't have a statistical comparison. But as it stands, it would be argued that the placebo group had worse fatigue symptoms after 24 hours of being awake. And we can see that for the creatine group versus the placebo group right here, 3.04 versus 1.69. Again, you want those to be decreased. For anger, at the 24-hour period, you can see creatine goes up to 0.15 from 0, and the placebo goes up to 1.11. That's, a 4 or 5-fold difference. So in this situation, you might argue, again, with no statistics to back it up, you might argue that creatine would see betterment. Tension, it seems like maybe half the effect of what we see with placebo. With depression, we see about a third of the effect that we see with placebo. And with vigor, maybe we see a slightly better amount of vigor with the creatine condition. But again, completely unsubstantiated by statistics, unfortunately. Okay, the next study is study 170. And this is actually from the same research group, but they wanted to extend the sleep deprivation. So I mentioned 36 hours earlier. That was incorrect. That was 24 hours in study 167. And now study 170, this is 36 hours. However, this was only in men, so 20 men, unlike the previous one, which was also in women. Is 21 years old, normal weight, generally healthy. And again, they don't indicate if it's randomized or not, so I don't know. It is placebo-controlled and double-blind, and they follow the exact same system as they did before. So if it's not broken, why fix it? So they supplemented with creatine for 7 days leading up, or placebo for 7 days leading up, and then they went through the experimental period. And here we've got our design of how they went about things. In this situation, they added stair climbing and step-ups and things of that nature. Probably pretty light activity. They can't be wearing people out completely, but maybe they want to wear them out a little bit to kind of increase that fatigue. And they took blood samples and salival samples along the way. So I'm only going to show you a little bit of data from this. They do look at a few other metrics. Again, they look at some hormonal differences like salivary concentrations of cortisol. I'll go ahead and tell you right now that there were no differences between the groups. And that is very strongly the case. Absolutely zero way that they were different if you look at the data. So I'm just telling you, if you want to look at the data for yourself, please feel free. Okay, so here again, this is the same research group. And yet again, they just refuse to indicate the statistical significance. They just kind of they make it as difficult as possible, unfortunately. So this is really just going by eye. So forward number recall, we got 6.7, and then at 36 hours, it's 6.9, 7.33 for the placebo. I don't think either one of these really changed that much. They changed about the same. Reaction time, we would want to see that decrease. So, or at least stay the same to have some sort of protective effect. And again, for this, there is maybe a little bit of a level of habituation, just like there might be for recall. So that's a potential confounding factor. But then that confounding factor would typically be applicable to both groups, not just the creatine group. So it may just be a wash. So the creatine group and the placebo group start out at baseline. Remember, this is before they were taking creatine, and then they end up doing measures again, and they see that, I would argue that potentially they see a decrease in both groups. So I would say that there's probably not that much of an effect in reaction time by creatine specifically. Then they did some balance tests, and they did end up reporting that there were improvements in balance in the creatine group. However, it doesn't seem to me like there was an improvement at the 36-hour period. It seems like it was only really around the 24-hour period and maybe the 18-hour period. But even that, I have a hard time believing, because the standard deviations are so wide. And they end up reporting that there is an improvement in balance in the creatine condition, but it's a little difficult to see, to be completely honest. So I'm a little skeptical. And then what was especially interesting to me is to look at the effort, because effort would be, you would expect that to at least be better in the creatine condition, especially from a muscular standpoint. But based on the exercise modality that they used, it seems like effort, or the perception of effort, actually increased about the same for both conditions. So there was really no real improvement between the two groups. Now that might be because of the type of exercise that they do. They do largely aerobic exercise as opposed to doing more anaerobic or lifting weights and things of that nature, which probably would have started to create that distinction if they had to do X amount of weight for X amount of repetitions, and then they had a hard cutoff. Maybe for the same number of repetitions, they might have seen an improvement with the creatine condition. But as it stands, they were doing a modality that isn't as conducive for creatine's benefit. So I would argue in this situation that we're not seeing any sort of strong evidence that creatine has a positive effect. Now, why might that be? Remember, we're looking at the same ages, we're looking at the same relative health of individuals, we're looking at the same research group. So, and the only real improvement, according to the researchers, was in measures of balance. So I would maybe chalk this up to the fact that this study had women included as well, which may have led to a particular effect that we don't see in men only. But it also, what surprises me a little bit in that scenario is that with women, you have maybe 25% of the sample or 20% of the sample that's women. So how much of an effect is that actually going to have as an overall effect? Kind of tough to say. So I'm a little bit disappointed, a little bit, I just don't, I'm not saying that the results aren't the results, but the heterogeneity, we'll put it that way, between the two groups or the two studies leaves me wanting certainly more research. But it's slightly leaning towards creatine having a benefit in sleep deprivation states. Now, finally going over a high stress situation before we actually get into dosing and whatnot. So for high stress situations, we can look at study 168. This is public funding. And for this, they recruited 15 men and women, and it was majority men. So 5 women and 10 men. They're 31 years old. They were generally healthy. It was randomized. It was double-blind and placebo-controlled, which is really great. 7 days plus experiment, again, following the similar style. So 7 days of supplementation with creatine, and then they did the experiment, and they did a long washout period, which is really nice to see because this is a crossover design. So the creatine group will end up consuming creatine for 7 days, undergo experimentation. Then they'll stop creatine supplementation for 5 weeks. Then they'll go 7 days on the placebo, and then they'll be tested again. And the placebo group, on the other hand, will go 7 days on placebo. Then they will be experimented on. Then they go 5-week washout, and then they switch over to creatine. Now, of course, they don't know that they're on creatine or placebo during this entire time. Okay, so this is the general organization. This is a wickedly cool experiment, to be completely honest. I'm not going to go over every single aspect, especially since some of this is areas where I think I would probably need to consult somebody that actually knows more about MRI imaging and MRS imaging. So I'm going to tell you what little I know and what I was able to pick up from some of the research that I did. So in this scenario, what they're doing is they're actively depriving these individuals of oxygen. So they're reducing their oxygen consumption by a certain amount. It's like 20% or something like that. And then they want to know, as they're oxygen deprived, how do they perform on cognitive tests? It's as simple as that. So the first question is, if they supplement with creatine, do they see an improvement in brain creatine concentrations, which is a pretty important question. If you're going to be supplementing, do you even see creatine reaching the brain? And the answer was yes, but depending on the measure. So if they looked at the whole brain matter, then yes, the effect was yes, there was an increase in the creatine consumption groups, which is CRM versus placebo. However, when they normalize it to the amount of gray matter, I believe it was gray matter that they normalized it to, or a certain type of brain tissue, then suddenly the effect was lost. Now, I would argue in this situation that's probably because they don't have the power to actually determine that effect, because it seems to me like we're very close to what we see here. But they maybe needed one or two more people, just a few more data points to actually be able to tease out that difference. So as it stands, I would say that there's probably still an increase. This p-value may be like 0.07 or 0.1 or something like that, and they just need a few more individuals to be involved in this. But as it stands still, they, at least in the totality of the brain, they did see an increase in creatine. This is a measure of what happens to their blood O2 levels, their blood pressure, and their heart rate. So this isn't necessary. The most important aspect here is, do we see changes in blood oxygen levels? That's the most important. The other stuff is just kind of fluff. It's an additional information that's intriguing, and it's cool from a physiological standpoint, but it doesn't actually give us any information on the effectiveness of creatine or anything like that. So when they deprive these individuals of oxygen, what happens to their oxygen saturation? They go from, like, let's say 99 or something like that down to around 80%. So there's a definitive drop in oxygen saturation. So that would indicate that they're oxygen deprived. So do they see any changes in blood pressure? The point is no, there's no change in blood pressure. However, there is an increase in heart rate. So if you were to think about it, why would there be an increase in heart rate? This is always a really interesting phenomenon that I like to throw out riddles every once in a while of why there might be some physiological change. That why do we not see a change in blood pressure, but we do see a change in heart rate when we see a drop in O2, in oxygen? Well, the reason for that is because the body's trying to compensate by increasing the amount of blood flow so that what little oxygen is available gets snapped up as quickly as possible by the tissues. So it's trying to compensate in that regard. And I'll bet one other area that they're probably compensating was increasing their draw of air. They were probably breathing a little bit more heavily as a result as well, trying to suck up as much of that oxygen as possible from the atmosphere into the bloodstream. So, and I'll bet you if they had continued doing this for several days, that they would start seeing creep upwards of red blood cell count, because the body would then be trying to compensate by trying to increase the number of areas, the number of cells and number of proteins, hemoglobin specifically, that can bind the oxygen that is available. Because again, the body doesn't know that you're actively trying to deprive it of oxygen. And it's a little bit similar to, what you might find when you go to a high altitude kind of situation. So you get greater expression of erythropoietin, EPO, and then you get greater levels of red blood cell formulation. And that's probably because of this drop in oxygen, and you see an increase in heart rate. Now, eventually you might see an increase in blood pressure as well if you start pumping more of these red blood cells into the system. But the big point here, the real takeaway here for our purposes beyond nerding out, is that with both conditions, you see a significant drop in oxygen concentration in the blood, indicating that there's problems with, or they're actually oxygen deprived. Now, there's a number of different tests like complex attention, executive function, cognitive flexibility, and then they've got a bunch of other different tests here. So for these, looking at p-values here, they're looking at just what's the effect of, is there an effect of if you deprive these people of oxygen? So this is not telling you about the effect of creatine or placebo. It's just looking, is there an effect of oxygen deprivation? And the answer is yes for a lot of these. So if you're looking at alertness, there's a drop in alertness. When you're looking at complex attention, there's a drop in complex attention. There's a drop in their SAT scores. So the error rate, I suppose I should say an error rate increase, so not a drop, but an increase. And the CPT, which is a continuous performance test, so the CE is the number of errors that they make. So the higher, the worse. We see that there is a worsening with hypoxia. You start to see some of them start to trend towards statistically significant. You have others like SAT errors as measures of executive function. You have, let's see, what else? SAT errors as in areas of cognitive flexibility. So they have different types of tests that they do that are related to SAT. Let's see, what else? Neurocognitive index, so composite memory, psychomotor speed, and reaction time were all affected negatively by the lack of oxygen, and complex attention was also affected. And cognitive flexibility started to trend towards significantly different. So there's a number of different metrics related to cognitive function that are affected by hypoxia. So that's really fascinating. Probably not shocking, but fascinating regardless. Now, in terms of the actual effect of supplementation, the effect that creatine has, so does creatine lead to some sort of modular effect? The answer is yes. Not on all of the metrics, but looking at, for example, complex attention, there seems to be an improvement, or at least an ability to bolster or to maintain complex attention. The reason why I say that is if you look at the baseline values, 93.7, and then you separate them out, creatine is still at 86.4, so a drop, but not nearly as much of a drop as the placebo. That went down to 70.7, so a really noticeable drop in complex attention. Then another one is looking at the CPT, so the error rate. We see that there's a reduction compared to the placebo. So the creatine group, yes, there's a slight increase in errors, but not anywhere near what we see. It's almost 3 times more in the placebo. Now, of course, the effect size, or the magnitude of the effect, the difference of the effect, I'm saying 3 times more, but it's really just 2 more errors. It's not like 20 or 30 more errors or 200 more errors. So, would this stack over the more tests that they do? Probably, but I don't think that the effect is like you'll be a genius if you take creatine or anything like that. It's very subtle, small effects. Then you have some of these that, trend towards significance. Again, executive function is another one of these. Cognitive flexibility is another one of these. Let's see, neurocognitive index, looking at, which is a combination of a lot of these. Complex attention, we see that there's an improvement. So I'm not saying that creatine is, there's maybe in cognitive flexibility, there might be a slight improvement. I'm not saying that creatine has massive effects. It certainly doesn't, but it seems like it lends itself a little bit to improvements. And we can look at these metrics. So this is the looking at the delta between these two conditions. And we've already seen these. So this complex attention, executive function, cognitive flexibility, and this neurocognitive index, which is the 5 that we talked about earlier: composite memory, psychomotor speed, reaction time, complex attention, and cognitive flexibility. So And they've separated out some of these individually. I'm assuming they did that because these are the ones that were actually statistically significant. And this grouping of them was probably driven by some of these effects. I'm not sure if that's why they did it, but I'm guessing that's the case. So for creatine, you want these to usually stay around baseline. So remember, these are these are normalized to baseline. So the further away you get away from baseline, that means the more of a difference that you have from baseline. So before they went through hypoxia, the loss of oxygen. And what we see is that with the creatine group, we still see some losses in complex attention, but nowhere near what we see, or it's kind of hard to say because there's a standard error that's really wide here. But at least it's statistically significantly different from the placebo group. We also see that for executive function, that's largely maintained. Same for cognitive flexibility, and there's maybe a slight decrease in the overall neurocognitive index, but it is statistically significant compared to the placebo group. Okay, and I think we're nearing the end. Yeah, we're nearing the final stages. So looking at this study, which was all in women. This was study 169, unknown funding, but some of the researchers have industry ties, sponsored research. This is misspelled. Industry sponsored research, as in they were given money by creatine supplement companies to do research on creatine. This particular study, I don't know, obviously it's unknown funding, and some of the researchers had no affiliation to creatine groups, but I wanted to disclose that regardless. For this, it was 26 women, 25 to 26 years of age, normal weight. They were randomized, double-blind, and placebo. This is a 4-week trial as opposed to the shorter studies that we looked at for 7 days. Creatine supplementation and placebo group, and we've got a parallel design. This is how they ended up allocating everyone. So they ended up having 13 in this group and 13 in both groups, and they ended up losing 1 on follow-up due to flu. To actually succumbing to the flu, unfortunately, not dying, but ending up having the flu, not because of creatine or not because of the placebo, but just because they got the flu, so they had to drop out of the study, which is completely fair. And so we've got 13 individuals in both groups. And that's how we end up with 26 in total. These are the baseline values. It doesn't tell you a whole lot of information, but they're 25, 26 years old, no differences between the groups. Body mass index, if you were to calculate this, it'd be roughly about normal. And then they had training experience. So they train Muay Thai, they train kickboxing for 2 to 3 years, something like that. Not terribly informative, but it's a little bit of information. Okay, now here we're going to break things down by these are the absolute values and here are the delta values. So they end up reporting absolute values so you can compare them, and then they end up they run statistics on the delta, so the actual difference between the creatine pre versus post and the placebo pre versus post. So they subtract those numbers and then they compare them against one another again. And we've got a number of different measures. Again, for a lot of this, it's reaction time based, obviously because they're doing sports, they're doing some sort of exercise, training, martial arts training. And then they've got a number of different tests, a Corsi block test, differentiation tests. Again, I'm not an expert in all these different measures. What I look for is, are there differences between the different conditions? Is it more memory-based? Is it more reaction-based, reaction time-based? Is it more executive function in terms of can you make a decision, a correct decision on a topic? And overall, so the way it functioned, I suppose I should explain this, is they had the participants come in after consuming creatine or placebo for X amount of time. Overall, the study was 4 weeks. And they then underwent a training session and then immediately afterwards had these tests performed. So this is a stress condition in a way, not like the hypoxia, but in a way it's related to just, high levels of activity and then immediately taking a cognitive test. And we can see that the only differences there are only a few different differences that we see. So creatine seemed to be better overall for reaction time, but we don't see any sort of big differences in terms of like memory tests or anything like that. So the only real differences come from the creatine group, pre versus post. But then they, like I said, they take these values, they subtract them from one another, and then they do a actual comparison between the two deltas. So looking at reaction time versus the placebo group, we see that the reaction time is obviously lower for the creatine, but we already knew that because we can also look at the absolute values. So I don't know exactly why they decide to run more statistics on this, maybe to show how really close they were getting to statistical significance. I don't know. But what they ended up showing was that there's a trend towards significance compared to the placebo group, and that's for both the reaction times. And we also see that there's a trend towards significance in some of these other measures that measure more of accuracy, like the arrows in the same direction test, where you see that there's reduced time versus the placebo group. Again, it's just trending towards significant. And I guess you could say correct answers as well tended to be improved in the creatine versus the placebo group. So some mild benefits, again, of creatine. We're not talking about becoming a genius when you start taking creatine by any means. Okay, so some other considerations here is that creatine seems to express most of its benefit in less than ideal scenarios for younger individuals. So that's in high stress situations with exercise or the hypoxia, no sleep. That data was a little bit wishy-washy, and obviously aging. So we saw that it improves for people that are over the age of 65, regardless of if they're in a stress situation or not. And number two, creatine may also be beneficial in highly cognitively challenging tasks, not just simple memory. So something that I determined, or what I looked into several years ago, which isn't quite as obvious here, is that if you put the brain under a high amount of load where a person really has to think and figure out a puzzle that is really challenging, it seems like creatine may be a benefit in those scenarios. But if you do the exact same test, but the puzzle is really easy and they just have to kind of piece together, then you lose that effect. So that creatine is only really beneficial when you have high stimulation of the brain, which makes a lot of sense related to the mechanism, because the mechanism is related to being able to excite the cells and to activate the cells and propagate signals at a faster rate, because you don't have to enter that quiescent stage and you have quite as readily. So you can excite the cells and then return back to a quiescent state very quickly. And you can only do that based off of the amount of energy that's available to actually be able to have that exchange of the ions. So if you don't have enough energy available, then you could end up in a scenario where you can't excite the cells nearly as much in a particular amount of time, because you're running out of energy, and therefore you're constantly ending up in this situation where the cell has to regenerate its energy to be able to fix its ion difference. And with creatine, you have this buffer, this added capacity for energy, which means that you can excite the cell maybe 12 times, when without creatine, you could only excite the cell 10 times in a particular amount of time. Hopefully that makes sense. And the only way you're going to actually get those cells to be continuously excited at maximum is if you are highly cognitively challenged. Okay, so let's return back to this meta-analysis. And here I found something really intriguing. So when we reanalyze the data, but we look at the rigorousness of the studies, if we remove the studies that lost more than 15% of the participants on the follow-up, so let's say 4 weeks later or 6 weeks later or several months later, and they were able to retain most of the participants in the studies, what we find is that there's a trend, a strong trend towards a significant effect in all age groups. Think about that. That would go against what I said earlier, but we don't have the granularity yet to actually indicate that. But it seems like more rigorously performed studies with a larger amount of data, that's reliable data, I should say, seems to trend towards, yes, there is an effect of creatine. But for now, I would feel more comfortable just sticking to the idea that creatine probably doesn't have that much of an effect on memory, because these are on measures of memory, remember, with this meta-analysis. Now, in terms of dosing, we're going to return to this meta-analysis again. This is a comparison of low dose versus high dose. So we're talking like 5 grams or 3 grams versus like 15, 20 grams. And we see that there's no effect. So if it's low or if it's high, it doesn't seem to make a difference. Again, unfortunately, they don't actually tease out the differences between the younger individuals and older individuals on high dose and low dose, which would be really intriguing. I would love to see the younger individuals, the data set with high dose, and the older individuals with the lower dose. That would be really intriguing to tell. But as it stands, there's no distinction between the two. And finally, the form in which they're taken. Unfortunately, this analysis isn't exactly great because they only have one study where it's in the pill form, and most of it is in powder form. And usually creatine is consumed in powder form. So, and the trend is towards there is an effect with the powdered form, but unfortunately it's also not statistically significant. Now, this isn't terribly informative just because, again, they're not actually separating out the older individuals because that's the only condition where they actually saw an effect. So they're not separating that out. So it would be actually more beneficial to separate out those individuals and then break it up by non-encapsulated form, which is powder form, or encapsulated form. Okay, so in dosing, studies used a range of doses and found effects using 3 grams up to 20 grams a day, so quite a lot. Its most common dose was 5 grams a day. And the type of creatine that was used was creatine monohydrate. I can go ahead and tell you creatine monohydrate is the cheapest, and it's just as effective as any other type of creatine, and sometimes more effective. So that's definitely the one to go with if you're ever interested in this. Obviously, check with your physician. So I still have a loose hypothesis that 20 grams might be sufficient. It might offer some slight benefits that 3 and 5 grams don't offer because those are more muscle centric. But as it stands, it seems like we don't have the granularity yet to actually determine the difference between the two. And if I go back here, this would actually indicate that I'm wrong, that 20 grams does not offer any sort of benefit that we can tell. But again, they're not actually pitting these two doses against one another. So we're not getting the actual differences between those two doses really compared against one another. It's just saying low dose in isolation, does that have an effect? High dose in isolation, does that have an effect? So I'd like to see more data, but that's honestly always true. So creatine supplementation likely improves other indices of neurological function, at least in the high stress circumstances. I'm a little bit less convinced by the sleep deprivation studies. We definitely need more studies on those to really tease out why there's some disparate results, and seems to improve metrics beyond memory in younger individuals. Remember, all the studies that we looked at, those individual studies, were all in younger individuals. So clearly creatine does have a benefit in younger individuals, but the memory effect seems to be the strongest in older individuals and may not be apparent in younger individuals. And creatine monohydrate is the type, was commonly consumed at 3 to 20 grams a day, and the most common being 5 grams a day. Okay, that was a lot of information. This analysis extended far longer for the insiders. So I realize there's a lot of confusing data, and frankly, there's a lot more data that we need. We need, I'd say, like 20 more studies on creatine, and I think maybe we'll start to get a little bit more of that granularity. But as it stands, creatine is beneficial to the brain. a little bit less so in certain individuals, like younger individuals, but it seems to be high benefit for older individuals. And it's probably also offers some small benefits for younger individuals in terms of like reaction time and decision making and things of that nature in stressful, like actual physiologically stressful situations. Hopefully it was informative, and I'll hope to speak with you in the near future. Have a good one. Bye.